So our final speaker of the morning is Dr. Teitel, who is a professor of medicine at the University of Toronto and staff physician in the Division of Hematology and Oncology at St. Michael's Hospital. He's the medical director of the Hemophilia Comprehensive Care Program at St. Mike's, which is the largest hemophilia treatment center in Canada. He's also medical director of the hospital's clinical program in specialized complex care. He obtained his MD degree and residency training at the University of Toronto and was a research fellow at Harvard, where he worked on novel assays for the detection of coagulation activation. His clinical and research activities are focused in the field of hemophilia and other bleeding disorders. He is also a past president of the Canadian Hematology Society, the Association of Hemophilia Clinic Directors of Canada, and the Hemophilia Research Society of North America. And he remains very active in the HCDC as our permanent executive vice president and a member of the inhibitor, database, and factor IX committees. And he's going to speak to us today on gene therapy in hemophilia.
Thank you, Jason, and thank you to CHS and David Page for the invitation. It's a pleasure to be here with you today. And I'm going to talk about something that's exciting as exciting as the things we've heard so far today. So these are my disclosures, and I'll also give you a couple of disclaimers. First of all, with apologies to my friend Dr. Lillicrap, I am not going to do any gymnastics. I don't know how to wave my arms and simulate a factor VIII gene, so I won't do that. My other disclaimer is that I'm not an expert in gene therapy, and some of these concepts are very technical, and I apologize if they seem a little bit esoteric, but you know what? That's the world we're all going to live in, so we have to get used to this. So let's dive into it. And I'm going to give you some background first and then talk about some of the trials that are ongoing. So looking back in history, the first steps on the road to gene therapy occurred back in the mid-1980s when the factor VIII gene was first characterized, and here are two papers published in the same issue of Nature, and in the same year the factor IX gene was characterized. And these advances, which were huge advances in the field, quickly led to the production and very quickly led to the commercialization. Within 10 years of the genes being cloned, we had recombinant factor VIII and recombinant factor IX shortly thereafter. And that's an example of where these recombinant proteins are produced. And these set the stage ultimately for gene therapy. And it led me to make a very bold and prediction. I used to tell my patients in the 1990s, by the year 2000, we will have gene therapy for hemophilia. So those of you to whom I actually said that, I apologize. How could I get it so wrong? So why are we still now talking about early clinical trials for gene therapy in 2017? Well, to be fair, there actually were early and successful and promising trials of gene therapy. And some of you will remember these. This is one from Katie Mano, factor IX gene transfer with an AAV2 vector. I'll talk more about viral vectors in a few moments into skeletal muscles. This was successful, as shown by muscle biopsy, which showed that there was the transgene expressed in that muscle. And interestingly as well, 10 years after that study, one of the subjects died of unrelated causes. And at autopsy, they were still able to find in the same muscle expression of the factor IX vector. So that was a successful trial. Another one that some of you will remember was a hepatic artery injection, again, of an AAV vector that did show good initial expression of factor IX. You can see that in the red line here. But it was very transient as there was an immune response to the vector that eliminated the factor IX expression. So these were very promising. So what happened? Well, what happened was some terrible events. And here's one of them that some of you will remember, published in 2003, a serious adverse event after successful gene therapy for a severe immune deficiency syndrome. So this is a little child, little baby who developed fatal acute leukemia a year after treatment, successful treatment with gene therapy for X-linked severe combined immunodeficiency. This was a retroviral vector which integrates into the patient's genome, and this young child died of acute leukemia. And that was in 2003. The same year, some of you will actually recognize this good-looking young man. This is Jesse Gelsinger. Jesse Gelsinger died of a fatal systemic inflammatory response after he received an adenovirus vector infusion to treat this enzyme deficiency. It was a severe disease. It was not fatal, but 4 days after he received the gene therapy, he's the 18th subject in the trial, he died a very rapid death of inflammation. And these terrible cases, these terrible complications, stopped gene therapy in its tracks for a good 10 years until the experts in the field could figure out how to avoid these complications. And that's why really we saw very little about gene therapy, and we talked about other new therapies, and only now are we starting to hear about gene therapy. So even now, in 2017, there are only 3 genetic therapeutics approved anywhere in the world, all in Europe, approved by the European Medicines Agency. One of them, the first one approved in 2012 to treat a lipid disorder, another one to treat malignant melanoma, and a third one for immunodeficiency syndrome. And as last time I checked, there were fewer than 10 patients in all of Europe receiving any of these products. So there's very little gene therapy going on anywhere in the world. One of the companies, Spark Therapeutics, hopes to become the first company to have a licensed gene therapeutic in the United States, which would be for treatment of a hereditary form of blindness. And it's possible that this will be approved sometime this year. But so far, there's nothing in North America approved. So what are the things we have to consider, the variables when we look at potential for success in gene therapy? One of them is the vector. Now this is the way, in other words, that we deliver the transgene, the gene that we want to deliver, to a cell in the body. So I use that, the editorial we, the people who are experts in the field are very clever at making these products, but they can't really figure out a good way of getting it into the cell of interest, so we take advantage of nature. Nature has a perfect way of delivering nucleic acid to cells, and it's called a virus. So we use viral vectors, take out their own DNA, insert the DNA of interest, and that viral vector knows what cell to go to and how to insert it into the nucleus of the cell. So in the sort of bold and asterisk shows you the vectors that have been, the vector that has been chosen for most of the trials that are ongoing for a number of reasons that we can't get into this morning. And the vector that's chosen is called AAV, which stands for adeno-associated virus. This is a virus which does infect humans but does not cause any discernible disease. And it's got a number of characteristics which make it very favorable for gene therapy. As far as the route of administration, clearly intravenous administration is much more palatable than injecting it directly into a muscle or into your hepatic artery or other sites. So that's the site, the route of administration that has been chosen. And finally, the target site. You can put the gene pretty much anywhere that has access to the bloodstream. The liver is chosen for most of the studies for a number of reasons. It is the only site in which factor IX is normally produced in the body. It is the major site in which factor VIII is produced. The liver is a large organ. It can take a lot of abuse. Some of us abuse our livers more than others, and come back for more. So it's a very resilient organ. So what are the risks and limitations? Well, one of them is something that I alluded to earlier, which is an immune response to the viral vector. Now, that immune response may be pre-existing or it may develop after therapy. So it may be pre-existing because, as I said, these AAV, for example, does infect humans. So some of us will have antibodies against the specific AAV serotype used for that gene therapy trial, in which case we would not be suitable for that drug because our bodies would reject the vector before it could get into the liver. So it might prevent initial treatment. The development of immunity after infusion can lead to an inflammatory response, which is usually self-limited and not very serious, unlike the response that was seen with the adenoviral vector in the Jesse Gelsinger case. But nevertheless, as an inflammatory response occurs, it may reduce the expression of the transgene. And finally, after receiving a certain serotype of a viral vector, you're going to develop immunity to it, and that means you can never be retreated with that specific vector in the future should you need retreatment. The next bullet point shows an important thing, and that is we don't know how long the effect is going to be. Gene therapy is still pretty new. The first report in hemophilia came out in 2011, so far so good. 6 years is good, but we're looking for even more. The approaches we're using so far are not suitable for young children, and largely because the liver cells in young children regenerate rapidly, they're growing, and the transgene will not express itself in new cells, which divide. We have to be concerned about possible germline transmission. We want to treat that patient. We don't want that transgene to get into genetic tissue that he will pass on to the next generation. We have to be, this sounds almost too good to be true, but we do have to worry about levels that are too high. We know that abnormally high levels of factor VIII and factor IX can be associated with thrombosis, so we don't want to overshoot with gene therapy. And finally, we have to really be concerned about mutagenesis, meaning cancer, if that viral vector delivers the gene which integrates into the DNA of the host, in which case it could activate a cancer-promoting gene. Now, why should that occur? One of the reasons that AAV is favored is because it's called a non-integrating vector. So how can a non-integrating vector be a cancer risk? Well, part of the answer you can kind of infer from the fact that I put non-integrating in quotation marks there. There's no such thing as a non-integrating vector. A non-integrating vector, as we refer to AAV, means it doesn't integrate very much. There's very little integration, but there is always some. It's like going to a restaurant and telling the waiter that you're allergic to seafood, and the waiter says, oh, don't worry, there's very little seafood in this dish. Well, a little may be enough. Now, it's a forced analogy, but you get my point. And so for a non-expert like me, I get nervous when I see reports like this. So this is from a paper in 2007, AAV vector integration sites in mouse hepatocellular carcinoma. So these are AAV-treated mice for a form of other disease, not hemophilia. 6 of 18 mice treated with that AAV vector developed liver cancer, hepatocellular carcinoma, compared to only 1 of 25 mice who were treated with bone marrow transplantation. And then a more recent paper, the bottom one, recurrent AAV2-related insertional mutagenesis in human hepatocellular carcinomas. This showed clonal integration of AAV in known cancer driver genes in a small proportion of human hepatocellular carcinoma samples. So these are cause for concern, but then came a lot of rebuttals. And there's a lot of words on this slide, but I think it's important to know how the experts interpret these data. So sorry, the print is a little bit small, but the first one, actually, this is the same author as the author of the second paper I showed you on the previous slide. And he says, this is Dr. No, who says, artificial infection of proliferative cell lines induces a different insertional profile compared to natural infection. Our study did not aim to challenge the safety of AAV vectors in gene therapy. The risk of liver cancer is probably very low if it exists at all. The next paper is actually an experimental study which characterized the integration of AAV in non-human primate tissues and in clinical liver biopsies. In other words, biopsies of human livers. Our results show the absence of genotoxicity of a systemically administered AAV in the large animal model and in human clinical context. And finally, there are several lines of evidence that contradict the assertion that AAV is associated with liver cancer. No cancer of any type has ever been observed or reported in human trials. And these are all true. So I give you all these just to tell you that we've got to be cautious, we've got to be careful. These are still experimental therapies. So with that, with all those cautions, let me tell you now about the current ongoing gene therapy trials in hemophilia. First, for factor IX, I'm aware of 4 ongoing trials for gene therapy in factor IX. The earliest one, the St. Jude University College London, the Nathwani study, first reported in 2011, the others all ongoing, and, and some of them still recruiting. And I'll show you some of the data. So this is the longest running study so far. First, as I said, reported in 2011. This is from their 2014 update. And this shows the 6 patients who had the highest dose of the gene, of the vector genome. And you can see that after an average, or a median rather, of a little over 3 years, these patients had an average, the factor IX levels are shown in orange, they had an average factor IX level of about 5%. So still doing very well, and some of these are many years after their initial infusion. And the green line shows liver function, little spike in some patients in ALT levels. This is the SPARK study, as presented by Lindsay George at the last ASH meeting, showing the first 7 patients treated. These patients were treated not with normal factor IX, but with a super factor IX, a gain of function factor IX called factor IX Padua. 3 of these patients are actually ours from our St. Michael's Hemophilia Treatment Center. And you can see that very short follow-up here. We're only talking about a few months, but you can see the levels are in the 20s and 30s% range of factor IX. And we're up now, I think, over 10 patients in this study, and everyone's still doing very well with good expression of factor IX in this particular study. Another study that's been reported, the Unicure study for factor IX. This is, again, a wild-type standard factor IX gene, intravenous administration, and again, mean factor IX levels when reported at the last ASH meeting were again in the 5% range, similar to the Nathwani study. What about factor VIII? Well, here's 5 that I'm aware of factor VIII gene therapy trials. Some of them are recruiting, others starting soon. And one of them was reported at the last WFH meeting. This is the BioMarin trial. And, you know, I told you earlier, we have to be concerned about very high levels. Look at some of these levels. These are the patients who had the highest dose of the AAV capsid. 2 of these subjects had levels over 150% of normal factor VIII. All right? I mean, we talk about prophylaxis and levels of 2%, 5%. One of these subjects had 270% factor VIII. That's higher than normal. It's not catastrophic, but it is of a little bit of concern. Now, finally, the last few slides, I'm going to talk about some newer therapies which are coming. This sounds like science fiction, but believe me, it's actually science. This is gene editing, even more sophisticated than replacing a gene. What if you can correct the deficient gene? So this would correct the defect in the existing alleles of the gene. It will allow you to maintain the regulatory context of the gene, potentially suitable for young children, unlike the gene insertion studies, and has a potential of danger, though, for off-target effects. In other words, you want to correct the defective gene. You don't want to go messing with normal genes in the genome. So, proof of principle. This is a study from 2011. These are animal studies, but this shows hemophilia B mice after intraperitoneal injection of a gene editing technique called zinc finger nuclease, or ZFN. And you can see that these mice expressed good levels of factor IX in their blood. And the arrow on the right shows that after partial hepatectomy, in other words, part of their livers were removed, continued to have good expression, which is important. So as the liver cells regenerate, they continue to express factor IX. That's why I said these techniques could be suitable for young children. And there actually is a study with this technology that is now recruiting in humans. This is the Sangamo Trial. So a zinc finger nuclease gene editing trial is ongoing in humans. Now, even perhaps more sophisticated than that technology is one that a lot of us have heard of. Don't ask me to explain it. I can at least say the word, CRISPR-Cas9. So this is an even more sophisticated and apparently easier technique for gene editing. And again, this is real science, not science fiction. So this is an article that came out in 2015, functional correction of a factor VIII gene using patient-derived, that is human-derived, induced pluripotential stem cells that were then corrected in vivo, or sorry, ex vivo, and infused into a mouse, and you can see that those mice who received those edited, corrected genes had better survival after tail clipping. That's the green line, so you can see more of those mice survived compared to the control mice. And this was just published online last week, another CRISPR-Cas9 study. This is from a Chinese group, and this is actually an in vivo gene correction in mice. And you can see the left-hand panel shows the factor IX RNA detected in their liver tissue, and the two slides on the right show factor IX antigen and activity detected in the blood of these mice. So, to summarize the last couple of slides now, some context. So we have gene therapy, an exciting and promising technology, good clinical trial results so far, but we have residual questions about long-term safety, long-term efficacy, and something that Jason alluded to a couple of moments ago, payment. So right now, all patients treated with these are on clinical trials. Well, after these become licensed therapeutics, who's going to pay for them and how are they going to be paid? And at the same time, simultaneously, while these exciting results are coming along, we have the things that my colleagues spoke about earlier in this session. Other new hemophilia treatments, longer-acting factor VIII and IX products that Shannon spoke about, antibodies simulating the role of factor VIII, rebalancing the coagulation system, subcutaneous, and potentially even oral therapies. We now have good animal studies showing the effectiveness of orally administered plant cell-derived factor IX, that is factor IX gene expressed in plant cells. So in this environment, I'll ask a question which I wouldn't even attempt to answer. What proportion of people with hemophilia will be eager to become early adopters of gene therapeutics when we've got these other exciting new protein therapeutics? So I'll leave you with that question, and I'll thank you for your attention.